Hi guys, by now you should have read the information, the 2 pages from Khan Academy that I posted about Gregor Mendel and his experiments with pea plants and the beginnings of our study of inheritance. Today I'm going to show you what to do, how we use Punnett squares and pedigree charts for studying genetics and tracing traits. So let's start with a Punnett square. A Punnett square, you put both alleles of each parent in the boxes. So you've got parent 1 alleles here and parent 2 alleles here, and then you take one allele from each parent to fill in the grid, just like this. You get this allele, and it makes that. So let's talk about describing this. Parent 1, we're going to call T tall, and it is dominant over the recessive short, and we're using the lowercase t to represent the short. Parent 1 has 2 dominant tall alleles. Because they're the same, it is homozygous, and because they are dominant, it's dominant, so we call the parent homozygous dominant. Parent 2 has 2 recessive alleles. They're the same, they're recessive, homozygous recessive, and all of their offspring each got a dominant allele from parent 1 and a recessive allele from parent 2, different, making them heterozygous. Let's look at a Punnett square where we take 2 heterozygous parents and cross them and see what their offspring look like. So both parents are heterozygous. When you start to fill this in, you get some differences. In that last one, all the offspring were expected to be the same, have the same genotype. And in this one, we've got some different genotypes. We've got 25% of the offspring with this homozygous dominant. They got dominant alleles from both mom and dad. Then we've got 50% of the offspring from these 2 boxes that are the heterozygous. They got a dominant allele from one parent and a recessive allele from the other parent. And then our last 25% of our offspring are the homozygous recessive because they got the recessive alleles from both parents. The question, though, is what are the offspring phenotypes? We know that these heterozygous and the homozygous dominant are going to show the dominant trait, which means these 3 would be tall. And this one, because there are both recessives there, the recessive allele is the one that shows, so this one's going to be short. So you would call this 3-to-1 tall to short. Let's look at pedigrees now. This is a way to track traits through a family. You can figure out genotypes by looking at the phenotypes through several generations. So here's a sample pedigree. In all pedigrees, a square represents a male and a circle represents a female. And you can show relationships. This is a married relationship when you put a bar between 2 people, and then coming off of it are their offspring. Now, if they only had 1, we would put either the square or circle at the bottom of it, but because they have 4, we put this line with the other small lines coming down. These are their 4 children. And then they each got married. You can just follow the lines. We are going to look at tongue rolling. Tongue rolling is a dominant trait. If the person has that trait, their phenotype is that they can roll their tongue, their circle or square will be colored in. If they do not have this trait of interest, their circle or square will just be an empty circle or square. So let's look at Grandma and Grandpa. Grandpa can roll his tongue, Grandma cannot. They had 4 children who can roll their tongue. Each of those children married someone who cannot roll their tongue, and then they had a mix of children with different traits. When I do this, I start with the recessive because if they're showing the recessive trait, we know they have to be homozygous. So I will always take everyone without a dominant trait and fill in the 2 recessive allele, the code for that, which in this case is the lowercase r. So all of the people who cannot roll their tongue get this lowercase r. For everyone who can roll their tongue, well, we know they have to have at least 1 dominant r. So I will go ahead and stick 1 capital R to represent the dominant allele in each one of those boxes. And from here, we really kind of have to do some extra thinking. Now I'm thinking if I look at their parent, then I can see, are the parents dominant or recessive? What do they look like? And each one of these children, got the 4, and all 6 of the grandchildren who can roll their tongue have a parent that has the dominant trait and a homozygous recessive parent. So we know every single one of these had to get a little r, or that recessive allele, from one of their parents. So we can go ahead and fill those in. Grandpa's a little different though, because we don't have information on his parents. We don't know if he's dominant or recessive. We might feel inclined to go ahead and say, oh, all four of their children have the dominant allele, so he must only have the dominant to give. He must be homozygous dominant. But that's not necessarily true. Those percentages reset every time you have a baby. So he could be heterozygous, and it just happens to be that this is the allele that showed up in all four of the kids. So because we don't know, we just put a little line here, and that is a perfectly acceptable answer. If I ask you, what is the genotype of this grandpa? You would just want to put this. Let's look at a recessive Punnett square. Sorry, pedigree. This pedigree is for something like sickle cell anemia, which is a genetic disease and it is recessive. You have a couple here. Dad does not have sickle cell anemia, mom does. They have kids who do not have sickle cell anemia. We know that they have to be heterozygous because they had to get the recessive allele from mom. And then they each marry someone who does not have the disease, and then they have children, and none of their children have the disease. It's a little weird here because these two cousins married each other, but it demonstrates really well some of the problems with inbreeding. They had three kids, and two of them have the disease. So we can follow these recessive alleles through all of these generations to find them here. So we know that these all had to be heterozygous so that that allele could keep getting passed on and show up eventually. So this was just some examples of how to use Punnett squares and pedigrees. You have a quiz on this that you need to take, and there's also an inheritance lab report you need to do. So there's discussion posts where you need to post your phenotype of a couple of different traits, and then we'll use class data to do that one. So good luck, guys.